Okay, so here we continue going to talk about this part of the lecture. Okay, so first of all, we're going to finish talking about the capsule. So what is a capsule? This is Klebsiella ammonia. In the lab, what I mentioned is Streptococcus ammonia. So you will see those are the large-shaped bacteria. Then they packaged by a very heavy, I would say it's a coat. What it looks like, it's like a coat, a heavy coat for a bacteria. That is composed by polysaccharides. That is called a capsule. And they are well organized and not easily to be dissolved, removed from the cell. And you use capsule stem. We talk about a capsule stem, which is negative stem, something like this. Or we can do a directive capsule stem using copper sulfide. Then we can do an observation under light microscope. Now the capsule is very important. First of all, it's a protection for bacteria, to protection from the stress environment, such as desiccation. And it's also going to be resistant to the phagocytosis, because for the host cell, usually they will generate a phagosome, and the phagosome will generate lysozyme to lyse the bacteria, and the capsule will be protected. Now, of course, it will be also penetrate to a little bit of the attachment, and the further will generate pathogenic material. That's for capsule. Now, ribosome. First of all, you need to know ribosome is the place for synthesis of protein. So that is called RNA translation. Ribosome has two units, which is 30S and 50S. When they combine, it's become a 70S ribosome. What does S mean? Which is called the Vandenberg unit. That is used to manage the precipitation speed of a protein. Now be careful. I have the very easy question before, which is a whole ribosome, which is 70S, is not 80S. Don't do a simple like add together. 50 add 30, that not equals 80. That's 70S, because that's S is a Vandenberg. Now, if you break down these two subunits, you will see under 30S subunits, there is a 16S rRNA. This is very important because under 16S rRNA, in their V6 and the V8 area, they are very conservative sequencing for all the microorganisms and for all the bacteria. That is where the 16S rRNA molecular technology come from. You may have heard about this if you do some of the research, undergraduate research with some professors. Now the 50S subunits, we have another small unit, which is 5S and 23S. Those all contribute to the components of transfer RNA, which is going there and then going through RNA translation. In detail, we will talk about in the examination 3. So that's for ribosome. Now nucleus. Now, first of all, you need to know as a bacteria, the nucleus is naked. Like we just mentioned, there is no membrane bundling system, so no membrane bundle. That's a location of chromosome and some of the proteins. And the nuclear gases, if it refers to the DNA, that is usually a closed circular, double-stranded DNA. You know it's semi-conservative and super negative, super coiled. Okay, so it's super coring aid in the folding. Folding, we call it modification, modify after the DNA replication. So that's for nuclear. Plasmids. Now, regarding the plasmids, this is, you can understand, which is a small sequencing, small size of the DNA sequence. They could be circular, could be linear. They can be exist independent by chromosome, or it can be integrated into the chromosome. So either way it works, and it's very flexible. And if it's integrated into the chromosome, it's called episomes. Now it is very important. Why? Because the plasmids, although it's very small size, sometimes it's 10 to 30 base pair, it usually will carry on very important information. So for example, these days, we are talking about antimicrobial resistant or antibiotic resistant or drug resistant. Where it comes from? Usually the plasmid carry on those antimicrobial or antibiotic gene become a resistant plasmid. So we call it all plasmid. And then once it goes to the environment, and some bacteria can eat it, can take it into the body. So we call it a conjugation, transformation, or conjugation, translation, or transformation. Then they can be taken by other bacteria. Then the other bacteria, once they have those all plasmid, it will also become drug resistant or even multi-drug resistant. So that plasmid, if they carry some of the certain gene, it's going to be very functional. And they can be classified based on the existence, spread, and the function. So make sure you understand that the plasmid, which is an extra chromosome DNA, small size, small sequencing, can be relied on or independent of chromosome. Sometimes it's play an important role for bacteria to survive in the environment, such as all plasmid play important role for antibiotic resistant. Okay, now we're gonna move on to the external structure. Now we have a cell envelope. So we talk about cell membrane. Then we're gonna talk about the external structure. We're gonna talk about the villi, fibril, and the flagella, okay? So let's talk about the villi first. This is a fibril or villi. That's a very short thin hair-like appendages. They are a little bit for motility, not really too much for motility, and help for the attachment to the surface, but the more important, it is for DNA uptake. Because they have some of the villi, we call it a sex phyllis. They will bring a bridge, connect the bridge between the two bacteria, then transferring the plasmids from one to the other. This process, we call it conjugation. C-O-N-J-U-J-A-T-I-O-N, called the conjugation. That's one of the methods for transferring genetic information from one bacteria to another bacteria, rely on sex villi. So villi, really important, is for transferring genetic information, little bit for motility and attachments. Now flagella. We talk about the flagella. We also have a flagella stem, and we will talk in the lab a little bit. Bacteria has flagella. Flagella is composed by a protein called flagellin. So it's very soft. It is very important for bacteria because the bacteria needs to do movement. And we will have another slide to talk about that. Now, based on the location of the flagella, we classify bacteria into 4 categories, which is monotrichous, only one flagella. Amphitrichous, one flagella at each end. I don't have a picture here. Lithotrichous, which is a cluster of flagella at one or both ends. And the petrotrichous, which is flagella cover on the surface. So give you some more examples. Pseudomonas, which is a sporage bacteria. We see only one tail, one flagella on one end. We call it monotrichous. Second one is amphitrichous. Amphitrichous kind of like a Staphylococcus aureus, but we didn't have it. It's have a little bit, also a little bit of mobility ability. Lithotrichous, sparium. This is a sparium. You see one cluster on the end. But always remember, we have one bacteria, very important, have a very strong moving ability called Proteus vulgarius. This has petrotrichous flagella, which is covered on the surface. This bacteria has so strong moving ability, which is too strong, cannot even form colony on the plates. On the plates, you won't see a colony, you won't see only a cluster. That's called a swamp. Okay, so that is for the flagella. Now, for some of the bacteria, remember Treponema pallidum? This is a syphilis, called syphilis, which is a spirochetes. I say it has asexual filaments. Okay, it's moving, have a very strong moving ability. We call it a corkscrew-like movements. These type of spiral sheets do not have a real flagella. We call it endoflagella. So kind of like a flagella, but not really a flagella. Now, why flagella is important for bacteria? Because bacteria needs to do moving, especially when they have a chemical signals. The chemical signals sometimes could be bad, could be good. They have the signal system in their cell membrane. We call it stimuli. So some of them will trigger bacteria to live away from the signal. Some of them will cause bacteria to attract into that signal. That all rely on the moving. The moving is rely on flagella. Now, here, regarding the sensor, the stimuli to trigger bacteria move forward or move away from the chemical, we have a terminology for that called chemotaxis. So always remember, chemotaxis is round trip for bacteria. Like you buy a ticket, not only you go there, you also come back. Okay, that's a round trip for bacteria regarding a chemical stimulus. Now, last slides, we're going to talk about the endospore. We will do endospore stem in the lab if we had the opportunity, but I also going to draw on the blackboard if later on in the lab section. So I'll keep it a little bit simple here. So first of all, you need to know there is a two bacteria has endospore, two species name, Bacillus and Clostridium. Bacillus, we have Bacillus subtilis, Bacillus cereus. Clostridium, we will talk about for Clostridium difficile, tetanus, perfringens, and botulinum. So those are the four. They are really is in a very harshful environment. They form this very dormant structure. This is called sporation. But when the condition changes, this sporation could become a normal bacteria. That's called germination. Now, you look at the structure of the endospore. Something very similar for bacterial cell wall. You will see an exporium, the coat, the plasma, the cortex, which is composed by polysaccharides, very similar. But why endospore can live in a very stressed environments, for example, high temperature? Because inside, there is a very special structure called calcium diproconic acid and very small soluble protein. Those two, especially calcium diproconic acid, CAD, we also call it. That's play an important role for environmental stress. Now, endospore, we can do endospore stain. The endospore stain usually can determine by the location of the bacteria of the endospore, because the location is usually genetic dependent. Okay, so based on the endospore stain, we can see the endospore could be in the center, could be at the terminal, or could be in the middle between center and the terminal. We call it subterminal. And their location is genetic dependent. Now, another thing I also want to mention is that endospore is generated by Bacillus clostridium in very stressed, unfriendly environment. When the condition changes, it will be changed. So endospore do not have reproductive function, which means the mother cell has endospore, does not mean the daughter cell will naturally has endospore. They have to be both at unfriendly and stress environments. So this is endospore. Okay, so here we end our section, which is our lecture 3.